participate in talking about what works and what doesn't work in a class, then you're really letting other people help mold what happens. So I think it's just as important to talk about what works as well as what doesn't work, so that hopefully the good things get reinforced and the bad things get washed out. So I do, again, thank you for your comments and feedback on that. Okay, so I apologize. As I said, I brought enough cards, but we didn't get them all. Some people took too many. But what I will do is, as I said, you can come up to my office and pick up a card if you wish, or if you want to wait till Friday, I will bring more blank cards on Friday so that you can have a card for the examination. I will remind you, I put an email out because I had several questions on it. I will remind you that the cards must be completely 100% handwritten, okay? So no printed cards, no cards with something glued on it like a figure or something like that. You can put figures, but you have to draw them yourself. So anything that's on the card has to be handwritten by you. You can use both sides of the card, et cetera, but that's the deal, okay? So I've seen some very innovative cards and some very cool things. I've told the story, I don't know if I told it in here now, but I had a young man a few years ago who decided he could double the capacity of his card, okay? And he actually did. Now, I don't let this happen anymore, so I'll warn you in advance. So he doubled the capacity of his card. And what he did was he wrote the card in red ink, and then he wrote the card over that in green ink. And then he brought in red-green glasses, and it worked! I mean, I practically gave him an A just for creativity, you know. I flunked him, but I mean, you know, no, I didn't. But, so no, I won't let you wear red-green glasses to take the exam. But he was pretty creative. It was pretty cool. So he doubled the capacity of his card. And really, I mean, it was amazing. You put the red lens on, and you could see only the green. You put the green lens on, you could see only the red. It was just really awesome. So he doubled the capacity. Yeah. Okay, I'll remind you that the exam is on Wednesday at 6 p.m. in here. And hopefully everybody's ready for it. I will give a review session this weekend. Haven't set a time yet, but I will announce that. It will be in ALS 4001 like the last one was. And the main thing is I have to get it scheduled and get the building opened and so forth, and that takes a little bit of time and effort to do that. Okay, so we're nearing the home stretch, and depending upon how far I get today, we may or may not get near the end. I do have a surprise for you on Friday that's a little different that I hope that you will enjoy. And so there'll be reason to come on Friday. If I do finish early, we will do like I did for the last exam. I will take questions as sort of a mini review before everything finishes up on Friday. So there may be some opportunity to get questions answered for the material on Friday. Okay. Well, what I want to do now is go back to what I finished with sort of hurriedly last time, and that was the phosphorylase kinase activation. All right. So phosphorylase kinase, I will remind you, is the enzyme that puts the phosphate onto glycogen phosphorylase B to convert it into glycogen phosphorylase A. Okay? So that's what phosphorylase kinase is. Phosphorylase kinase itself exists in two different forms, an inactive and an active form as well. Okay? So phosphorylase kinase can be inactive, it can be active. And so you see the fully inactive form on the left, you see the fully active form on the right, and you see two things that contribute to activating this enzyme. Now, as I mentioned last time, there's no order to this. It doesn't matter which one happens first. Although if you're an exercising muscle, you can probably figure out which one's going to be activated first, and that's the bottom one. Because when muscle contraction happens, muscles control contraction through the release of calcium. And the release of calcium causes that contraction, which, of course, immediately causes a need for ATP. And the system is set up very nicely where the release of calcium also results in the binding to phosphorylase kinase of calcium to activate it. Okay? So the very thing that causes the muscles to contract activates, partly at least, phosphorylase kinase. That means that the fully inactive phosphorylase kinase is partially active, and it can start breaking down glycogen immediately. Now, it's not going to be fully active until phosphorylation happens, but phosphorylation is not going to happen until the hormones come and say, hey, look, this guy's running, and we're getting short on glucose. All right? So this gives an instantaneous activation. The phosphorylation can come later if you're an exercising muscle. If, on the other hand, you are a brain cell and you get the sense that, oh my God, we're in danger, we're going to have trouble, you start dumping hormone, or perhaps you're anxious about your exam and you're dumping anxiety hormones. This may, in some cases, happen first, in which case it's going to activate and partly activate, again, starting to release some glucose from glycogen, and when you take off running, there you're going to fully activate it and do this. So the net result of this, either way, or both ways, actually results in the conversion of glycogen phosphorylase B into glycogen phosphorylase A. Okay? So as I said earlier, we think always about glycogen phosphorylase B being the less active form, glycogen phosphorylase A being the more active form, and there are, of course, exceptions to that depending upon what's present allosterically. Questions on that before I go forward? Okay. Let's see. Now, what I want to do here is give you a bit of an overview in the body in terms of what's really happening, okay? So this schematic, which I think is not the best in the world, actually does help us a little bit to understand these different processes and how they tie together. So if my blood glucose levels are low and or I'm afraid, two things may happen. So glucagon is a hormone I've mentioned earlier. Glucagon is a hormone that's released by the pancreas, and it's released in response to low glucose levels. It doesn't have to be anxiety, it doesn't have to be something that scares you, but it's playing a very important role in recognizing when your blood glucose levels are low and telling the liver you better do something about it. Okay? So glucagon is a hormone. It is a peptide hormone. And when it's released, it goes to target cells. Glucagon will interact with liver. It will not interact with muscle cells. Epinephrine can interact with muscle cells. It can also interact with liver cells. So epinephrine can interact with both. If you think about it, that's kind of important because, again, epinephrine is saying we're in danger, we've got to get out of here. And so activating glycogen breakdown in muscle cells is a very important and useful thing. So depending upon what's happening, we have these guys being released into the bloodstream. They go to their targets. So in the case of glucagon and or epinephrine, they can go to the liver. And the liver, they stimulate two pathways. One pathway, of course, being the breakdown of glycogen to make glucose. The other pathway being the conversion of lactate to make glucose also. Lactate going to pyruvate, pyruvate going through gluconeogenesis. Where does lactate come from? Well, remember the Cori cycle. Lactate is coming from the exercising muscle cell. Now, students sometimes get confused about this because you say, well, look, here is a catabolic pathway, glycogen breakdown, and an anabolic pathway, which is the gluconeogenesis, going on in the same cell. Is that a problem? Something is on here. There we go. Okay. Is this a problem? Well, no. They're both directed at doing the same thing, which is the release of glucose by liver cells. They're not going in a futile cycle. To go in a futile cycle, you would have to have complete breakdown of one and the complete synthesis of the other. Right? They're converging at the production of glucose. That's what's happening with these pathways. And this is very important that they converge at glucose because glucose is the most important thing for cells to have. Most cells in the body cannot make their own glucose. They rely on liver primarily to provide them what they need. Your eye, your brain, totally dependent upon glucose coming through the bloodstream. And they can't make it themselves. Okay? It's why when your blood glucose levels get very low that you faint. Okay? Hypoglycemia. If you have hypoglycemia and you have fainted, the reason is your blood glucose levels have gotten very low. Diabetics, when they have problems with their insulin, what happens is they overshoot and the insulin gets too high, which causes too much uptake of glucose by the cells from the bloodstream. The brain doesn't have enough glucose to stay awake, and you faint. You pass out from that. We'll talk more about diabetes actually next term. Okay. At the muscle, what's happening? We see glucose that's been dumped by the liver into the muscle cell. We're seeing the other part of the Cori cycle now. Glucose going in here. And inside the muscle cell, what is epinephrine doing? It is stimulating glycogen to break down glucose into glucose 6-phosphate. Remember, it starts first as glucose 1-phosphate, then we have that phosphoglucomutase reaction. It's also stimulating glycolysis. Okay? So now we have, in this case, glycogen being broken down and glycolysis going on. They result in the production of pyruvate, which leads to the production of lactate, which now goes out in the bloodstream and ultimately ends up back at the liver. Okay, so that's a nice overview of the pathways between the two different cells and the effects of the hormones there. Questions on that? Quiet group today. Okay. I think I've shown this before, but if I haven't, that's epinephrine. Epinephrine is derived from tyrosine. It looks a little like tyrosine, and so there's a couple modifications that happen to it to make it do what it does. There's the sequence of glucagon. Glucagon is, as I noted, a peptide hormone. No, you don't have to know the sequence. But you'll notice it's a fairly short peptide. It only has 29 amino acids in it, which means it's relatively easily synthesized. It's also readily soluble in the blood as it passes through. And now what I want to do is turn our attention to what's called the regulatory cascade. Okay? You've seen part of this so far, and the part you've seen goes up to right here. You saw epinephrine binding or glucagon binding. And they both will bind to 7-TM receptors, and no, they're not the same receptor. If they were, muscle cells would also bind glucagon. But both glucagon and epinephrine can bind to specific 7-TM receptors. Glucagon only in the liver. That causes activation of a G protein. Specifically, it causes activation of the alpha subunit of the G protein, which causes it to bind GTP and to release the beta and gamma subunits. That activated G protein, in turn, you recall, hopefully, interacts with adenylate cyclase, activating it. And the activated adenylate cyclase produces cyclic AMP. Okay? Cyclic AMP, you saw earlier, allosterically activates protein kinase A by binding to the regulatory subunits and causing the catalytic subunits to be freed. The protein kinase A can now go do a variety of things. One of which you've seen here is it can activate phosphorylase kinase. Okay? Phosphorylase kinase, in turn, can convert glycogen phosphorylase B to glycogen phosphorylase A. Now, you might ask the question, why such a convoluted pathway to turn on glycogen phosphorylase when, in fact, what you want to do, why not just cut it short and make this, activate this, and bypass the whole system? And like for many things, I don't have any good answers for that, but I will argue that there are two benefits to having a system like this. One benefit is that this protein kinase A, which you see here, can be used to activate other pathways. It can be used to activate other pathways. We're going to see that protein, in fact, you're going to see today that protein kinase A will inactivate glycogen synthase, the enzyme involved in the synthesis of glycogen. Okay? This one system is allowing control of two different metabolic pathways, glycogen breakdown, glycogen synthesis. You've already seen a little bit about how this protein kinase A can be involved in controlling glycolysis and gluconeogenesis through the PFK2 and FBPase2 system. Okay? Those phosphorylations affect whether or not glycolysis or gluconeogenesis happens. So now this one system is affecting actually 4 pathways. And 4 isn't even the total number. There are many other pathways that are affected. Next term we'll talk about fat metabolism, and we'll see that protein kinase A in this system is also affecting fat metabolism. It affects cholesterol metabolism. Protein kinase A plays a very critical role. So we don't want to bypass some of these steps because they do relate to other things. The other argument I'll give for why this scheme makes sense is this is what we call a kinase cascade. If you look at my notes, sometimes you'll see I refer to this as the kinase cascade. This is a kinase, this is a kinase, and this makes this, okay? The kinase cascade, the cascading system allows for amplification at each step, okay? one cyclic AMP can activate one protein kinase A, which can in turn activate hundreds or thousands of these, which can in turn activate thousands or millions of these. So at each step of the process, we get amplification in a cascade. Now, the beauty of having such tremendous activation is that it can happen very, very fast. We saw the same thing happen in the clotting of blood. You saw that blood clots could actually form in the matter of minutes as a result of cascades that were happening there. Here, we're seeing that the control of glycogen metabolism can happen very, very rapidly as a result of an amplified signal through the kinase cascade. Anybody got that? Okay, questions? Okay, that's the last thing. Oh, yeah, question.
Would another benefit of having this cascade also be that it would allow for different levels of regulation?
So her question is, will another benefit of this allow different levels of regulation? And the answer is absolutely. Okay? So there's many different things that can happen. And what we discover is that protein kinase A, we're seeing this in a very simple scheme, can affect many other things that in turn may affect things here. One of the things I haven't told you about, for example, is that phosphorylase kinase actually binds to calmodulin. Okay? So you can see effects on calmodulin, you can see effects on the synthesis of calmodulin and so forth as a result of this here. So there are fine-tunings that can happen, many different controls that can happen. And those many different controls are very, very important because we tend to think about cellular decisions to be very simple. Do I divide or not divide? That's what I talk about in here. Okay? But then it depends on the circumstance. Do I divide because I have plenty of energy? That's a simple decision. But I've got plenty of energy and I'm a cell in the lens of an eye and I'm not supposed to divide. Oh, well then I've got to have a signal that I can modulate depending upon the place where it's at. So being able to control and have many levels of control for the decisions that cells have to make is very, very important. So you're absolutely right. Okay, other questions? Yes, Ryan. Say it again. Is there calcium in the liver to fully activate? Yeah, there's calcium in all the cells of your body. In fact, plenty. Calcium, as I sort of mentioned earlier, has to be sequestered because if it's not sequestered, then it's present in high enough concentrations, it could cause the chromosomes to precipitate. So it actually has to be sequestered. And so we talk about letting it out. We're not letting it all out at once. We're letting a little bit out at a time. But yes, calcium, and calcium is an important signaling molecule, so it's going to be present in virtually every cell. Okay. All right, so that's what I want to say about glycogen breakdown. I'm going to turn our attention now to glycogen synthesis. And just as we saw with glycogen breakdown, glycogen synthesis is almost as simple in terms of the reactions that are involved. You've seen a little bit of this so far, and the thing that you've seen so far is this guy right here. Last week I talked about, or the week before, I talked about UDP glucose. And you may not recall, but I'll remind you, UDP glucose is what we refer to as an activated intermediate. An activated intermediate is a molecule that has a high energy bond that uses the energy of that bond to transfer a portion of itself to something else. So it's a molecule that has a high energy bond that uses some of that energy to transfer a portion of itself to something else. Now, UDP glucose is the portion that gets transferred, of course, is the glucose that we can see here. And the reason that we have to have UDP glucose, I said in the last lecture. What is the reason we have to have UDP glucose? Why can't I just run the reaction backwards? The breakdown reaction backwards. What's that?
It's stuck at the Big Bang.
Well, Big Bang is in glycolysis. Why can't I make glycogen as the reverse of the breakdown of glycogen? It's the energy, absolutely. So remember that we have energy. The reason that the phosphorylasis was possible was because the alpha 1,4 bond had energy in it. And that energy has to be supplied by something. Here's the thing that's supplying the energy necessary to make that 1,4 bond. So we have to have an activated intermediate that allows us to add that glucose to make that high energy 1,4 bond so the glycogen itself can be synthesized. Okay? Well, this is what UDP glucose is. How do we make UDP glucose? Well, it turns out this is one extra enzyme that we have different from glycogen breakdown. And this shows the reaction that's being catalyzed in the synthesis of UDP glucose. The enzyme that you will need to know the name of, and it doesn't have an abbreviation, unfortunately, it's called UDP glucose pyrophosphorylase. Pyrophosphorylase. UDP glucose pyrophosphorylase. Now, why such a mouthful of a name? Well, look what we're starting with here. We start with glucose 1-phosphate. That's where you've seen this before. You've seen it in several pathways now. Glucose 1-phosphate combines with UTP, that's a nucleotide, uridine triphosphate, and it makes UDP glucose and PPI. That's pyrophosphate. That's the pyrophosphate that's produced by the pyrophosphorylase. Now, this pyrophosphate turns out to be very important. Whenever we see pyrophosphate occurring in a reaction, it turns out that it's just not a coincidence that it appears there. The pyrophosphate, if we count the phosphates, we've got 1 phosphate here, we've got 3 phosphates here. We have a total of 4 phosphates to account for. 2 of them are here, 2 of them are here. Okay? So in an accounting sense, we obviously have to make sure that they're all there. But in another real sense, this pyrophosphate is essential for helping this reaction to go forwards. How so? It turns out that cells have an enzyme that you've already heard something about before called phosphodiesterase. And phosphodiesterase will break down this bond between these 2 phosphates and release them as 2 independent molecules of phosphate, 2 PIs, as it were. Why is that important? There's an abundance of pyrophosphate in the cell, and this abundance of phosphodiesterase in the cell that's breaking this down. So as soon as the pyrophosphate is made, it's being broken down. And by the way, there are many phosphodiesterases. So just because we're taking caffeine doesn't mean we're going to affect this reaction, which is the one I talked about with the cyclic AMP is specific to that. Okay? Now, if we have plenty of phosphodiesterase, now what happens is this guy gets immediately broken down. We're pulling the reaction. This allows the cell to pull this reaction, to pull the formation of this high energy intermediate and favor very strongly the production of UDP glucose. The cell really doesn't want this reaction to go backwards. So phosphodiesterase assures us that this reaction will not go backwards, and it helps this reaction to go forwards. Now, you say, okay, well, here's one reaction where it occurs. Maybe it's not that big of a deal. Let me remind you that when you make DNA, you start with a triphosphate. What goes in is a monophosphate. That leaves a pyrophosphate out. One of the things that helps drive the synthesis of DNA and RNA is this guy right here. This is a very important molecule. Okay, so having a phosphodiesterase to break down pyrophosphate is very, very important. Okay, so now we've got our precursor, what we need to make glycogen. We now take that UDP glucose that we created before. We take an existing glycogen chain. And by the way, this requires an existing glycogen chain. You can't start making glycogen by putting together 2 different glucoses from UDP glucose. There's another enzyme that does that, and it's called glycogenin. Glycogenin. Glycogenin is an enzyme that starts the very first part of a glycogen chain. I'm sorry this is ruffling, but they lost the clip for this, and I had to improvise. Okay. So, this is pretty straightforward. What's created is an alpha 1,4 bond. We see the alpha 1,4 bond there, and the dropping out of UDP. Okay? So UDP is released. This high energy intermediate is donating this glucose to this growing chain. We see now this high energy bond that's been created here, and we see the release of UDP, which can now go back and be converted into UTP. Okay? Make sense? So So, what have we seen so far? We have seen that we need to have a UDP glucose pyrophosphorylase. We need to have an enzyme that does this. The enzyme that does this has a very simple name. It's called glycogen synthase, synthase. And there's one more enzyme that we need. What other enzyme do we need? What else do we need to have glycogen? We're missing something. Branching. So we have to have an enzyme that has a name that's about this long, and we're going to call it branching enzyme. Okay? So we have to have branching enzyme. And branching enzyme is simply going to create branches. Those branches are going to have alpha 1,6 bonds. And it's not overly complicated how it works. Okay? In fact, your book doesn't even, I thought, yeah, it doesn't show a figure for that. But suffice to say, branching enzyme makes the branches. All right, so again, the metabolism, that is the synthesis of glycogen, is very simple from an enzymatic point of view. The regulation is more complicated, but the regulation has a very cool logic to it for the most part. A lot of that logic you see right here on the screen. Okay? And you'll notice that it overlays with what we saw before. What we saw was that if we had glucagon or epinephrine binding to the cell surface receptors, they activated a G protein, which activated adenylate cyclase, which produced cyclic AMP, which activated protein kinase A, which activated phosphorylase kinase, which activated glycogen phosphorylase. Notice now, we overlay glycogen synthase on this. Protein kinase A, which will phosphorylate phosphorylase kinase, will also phosphorylate glycogen synthase. On this figure, the inactive forms of the protein are shown in red, and the active forms of the protein are shown in blue. Notice that the action of protein kinase A is converting active glycogen synthase into inactive glycogen synthase. Now, I want to point out something nomenclature-wise that is a little confusing. We used A to indicate the active form of glycogen phosphorylase. We also use A to indicate the active form of glycogen synthase. Okay, that's consistent, right? However, glycogen phosphorylase A has a phosphate on it. Glycogen synthase A has no phosphate on it. So the A and B do not relate to the phosphates. They're backwards between the two. Glycogen phosphorylase is called A when it has the phosphate on. Glycogen synthase has A when the phosphate is off. Conversely, when the phosphate is off, glycogen synthase is called B, and phosphorylase is called B. Glycogen synthase B, however, it has the phosphate on. I said it backwards. Synthase A, no phosphate. Phosphorylase A has phosphate. Okay. Synthase B has no phosphate. Synthase B over here has phosphate. Okay. All right. So the phosphates don't track. You see, I can get confused with it too. All right. Now, the beauty of this is a reciprocal regulation. The same thing is affecting a synthetic and a breakdown pathway in exactly opposite fashion. What's turning on the breakdown is turning off the synthesis. And that makes very, very good sense. We don't want to be making glycogen at the same time as we're breaking it down because we're going to be in a futile cycle. And if we're in a futile cycle, we are not going to be able to make anything productive, and we're going to waste energy. Okay. Now, the questions, maybe I should say, ask for questions. Questions on that before I go forward? Okay. Now, well, how is it that the glycogen synthase is itself activated? How is it activated? Well, it turns out that to be activated, glycogen synthase has to lose a phosphate. And you can imagine that losing a phosphate here will parallel losing a phosphate here. So as we make glycogen synthase more active, we make glycogen phosphorylase less active. All right? To remove that phosphate, we have to have something else happen. Okay? There's a different hormone that's involved in that, and the hormone that's involved in it is insulin. Insulin, and I'm not going to show you a cascading pathway for it. You can thank me for that. Insulin, and it's partly because it's a very complicated pathway, insulin stimulates the activation of an enzyme that we talked about before called phosphoprotein phosphatase. phosphoprotein phosphatase. Your book calls it PP1. Okay? Phosphoprotein phosphatase, as its name suggests, a phosphatase takes phosphates off of things, takes phosphate off of glycogen synthase, it takes phosphate off of glycogen phosphorylase, and it will take phosphate off of phosphorylase kinase, because all these guys have phosphate on them. So what's happening is the insulin signaling pathway is counteracting the epinephrine pathway. Exactly the opposite effects. And if you think about it, these are occurring for exactly opposite reasons. The reason being that this guy is trying to increase blood glucose. This guy, when we have insulin, is trying to decrease blood glucose. Okay. Well, how does phosphoprotein phosphatase itself become activated and inactivated? Here's PP1. Oh yeah, I told you it was involved. Here's glucagon or epinephrine. Protein kinase A, you've seen will activate phosphorylase kinase. You've seen it will inactivate glycogen synthase. Phosphorylase kinase will convert phosphorylase B into A and make it active. That's if we're coming down from above. And forget what it says about after meal or rest. It's a kind of a dumb scheme they've got here. If we look, on the other hand, where we have insulin binding, okay, because insulin binding is actually right here, right, after a meal or rest is down here, glycogen synthesis is required. Why is glycogen synthesis required? Because after a meal, blood glucose levels go high. Glucose is a poison. We want to get rid of it. So phosphoprotein phosphatase becomes activated, and it now reverses the effects that you saw before. Okay? So just another way of looking at that schematic that you saw before with the kinase cascade, but now showing the effects on the individual enzyme. Jenny?
So what's inactivating protein kinase A?
What's inactivating protein kinase A? Anybody know? What inactivates protein kinase A? Phosphodiesterase. That's what's breaking down the cyclic AMP. Right? So cyclic AMP is necessary for protein kinase A to be active, and we have enough phosphodiesterase that under normal conditions it has a very short lifetime. So it actually gets inactivated in that way. Okay, Laurie?
So is protein phosphatase the same thing that breaks down that dephosphorylates FPPase?
Is this the same one that dephosphorylates FPPase 2? And the answer is yes. Okay? So now again, we see that the pathways are connected. We see that the glycogen synthesis, glycogen breakdown ties into gluconeogenesis and glycolysis. And that's important. They're all sugar metabolizing pathways. And there is a logic to it. The logic is to either produce glucose or use glucose. That's what everything is directed towards. All right? So if we look at glycogen synthesis, that means we're using glucose. We can remember very easily that if we're using glucose, we must have too much. We must therefore be favoring the breakdown of glucose in glycolysis. They're both going to decrease the concentration of glucose. Okay. How is PP1 controlled? PP1 is controlled primarily by something that your book wisely calls an inhibitor. Many books call this PI1. In fact, I think some of my notes actually say PI1 as well. PI1 and PP1 are different things. PI1 inhibits PP1. The inhibitor inhibits the phosphoprotein phosphatase. How does this work? Well, normally we have a protein in our cells called G. And this is called GM because it's not a G protein, by the way. When we talk about G proteins that bind GTP, that's something different. They just happen to have the name G. The GM refers to the fact this is in muscle. We can have a GL for liver, etc., but this is GM. We're talking about muscle at the moment. When phosphoprotein phosphatase is active, it's bound to G, and it does its thing. The inhibitor is floating out here, okay? And epinephrine comes along. Epinephrine is going to favor the addition of phosphates through the kinase cascade. So if we're favoring the addition of phosphates, we don't want something active that's going to take those phosphates off immediately, or we're going to have a futile cycle, right? Epinephrine or glucagon stimulate activation of protein kinase A, and protein kinase A will do two things. One, it will phosphorylate GM so that it releases PP1, which makes it much less active. Second, the inhibitor will bind to PP1, and the inhibitor binds to PP1 after it too has been phosphorylated by protein kinase A. The bottom line here is protein kinase A is favoring the release of PP1 to make it less active, the phosphorylation of the inhibitor to bind and make it completely inactive. So now at this point, phosphoprotein phosphatase can't do anything. It can't remove phosphates from the various proteins, the glycogen synthase, the glycogen phosphorylase, and the phosphorylase kinase. Okay, I know there's a lot of steps to this, and I know there's sort of intertwined pathways. This is another example of where words aren't going to work for you nearly as well as if you sketch it out, and you sketch the relations between these pathways out. If you do that yourself, I can guarantee you it's going to make a lot more sense than a million words I'm going to throw at it. Okay? Much as I would like to think I'm very clear and coherent, writing it out yourself is going to make a lot more difference than anything I'm going to say about it. Okay? And what you will do when you sketch that out is you're going to see the consistency between these pathways. The consistency of one completely activating the breakdown of glycogen, the other one completely activating the synthesis of glycogen. Okay? How do we reverse this pathway? Well, one of the ways we reverse this pathway is, again, through insulin signaling. And there are other phosphatases that are activated upon insulin signaling that now take the phosphate off here, take the phosphate off here, and we go back in the direction of a fully activated phosphoprotein phosphatase. Okay. That's a good stopping point, I think. So I should have asked for questions, but I guess that means I won't have any questions at this point. All right. We'll see you on Friday. We will have a surprise on Friday, I promise.
Disorders or diseases that cause you to get both the insulin and the epinephrine pathways both being signaled at once?
No. You're dead. No clip. The clip is gone. Yeah. How did you do it? I threw it away. Yeah, what's up?
So the PKA, it uses ATP to activate or to make these two come apart, and that's because it binds an ATP molecule to. So
no, you've got protein kinase A. You're talking about protein kinase A? PKA?
Yeah.
Okay.
Yeah.
Protein kinase A phosphorylates. Okay, phosphorylates this guy, which causes this to be released.
Okay.
All right. So this guy's released. It's less active when it's in the release form. It's much more active in this.
Because the inhibitor can bind to it. And
well, it's actually in this state. It's much more active, bound to this.
Yeah.
So the release of this from this just alone causes it much less active.
Okay.
The full inactivation of it requires the addition of the inhibitor.
Okay.
Okay. Hi. Yeah.
Arl's phone.
Okay. What's up?
Just like what the concerted model